physiological mechanisms underpinning physiological and pathological aging by TMS and EEG, which, by the way, was the last contribution of Florinda, who developed this very neat signal based on TMS EEG for predicting which MCI patient eventually would turn to Alzheimer's disease 5 years later. It was really this small but really quite incredible study that she has done. And that was one of her last publications. So Andrea, it's all yours.
Well, thank you for the introduction. Yes, a lot for me, and it's really a pleasure to have this lecture in memory of Florinda, which was my first teacher. I would say I was a student when I started, and it's really a pleasure. I hope you see my slide.
Yes, it's fine.
Okay, thank you. So the outline of my presentation is this one. I will talk about 3 techniques that I learned from Florinda. So TMS, transcranial magnetic stimulation, EEG, and the combination of the two for studying physiological and pathological aging. So starting from TMS. So TMS, probably you know that TMS implies delivering magnetic stimuli from a coil placed over the scalp, which, according to the Faraday law, induces electrical current into the brain. And when delivered over the motor cortex, the stimuli activate the corticospinal tract and produce an EMG activity over the contralateral target muscle, recognized as motor evoked potential. So the amplitude of this potential is a proxy of the corticospinal tract excitability. So there are many techniques which could be applied by using TMS. TMS can be applied by single pulses, by pair pulses, so 2 pulses through the same coil, and by repetitive technique or pattern technique, which induces plasticity processes. So I will cover mainly studies focusing on single pulse technique and repetitive TMS techniques. So Florinda was one of the internationally recognized experts of TMS. Actually, she is a co-author of the latest guideline from the International Federation of Clinical Neurophysiology on non-invasive electrical and magnetic stimulation of the brain. And the first technique that I want to talk about is related to the first study of Florinda when she was a student. So this is the motor cortex mapping technique, implying delivering single pulses of TMS over a number of cortical sites and recording the MEP amplitude for each site. By doing so, on the right side, you could see that it's possible to draw a map of the cortical representation of the muscle that we are targeting. And in the triangle, you will see the hotspot, so the site that elicit the largest motor evoked potential. And in color, you will see in black color, in grayscale color, the site that elicit an MEP with gradually increasing amplitude. And so in this way, you will create the map, you will measure the area of the map, the volume of the map, and also the center of gravity of the map. So practically, how much the map of cortical representation is balanced or is shifted compared to the hotspot. So using this technique, Florinda demonstrated for the first time that in Alzheimer's disease patients, even in the early phase of the disease, there is an hyperexcitability of the motor cortex. But also, more importantly, she showed that the center of gravity of the cortical map is rearranged in patients because it's shifted anteriorly and medially compared to the control subject. And she interpreted, and this was quite a speculation at the beginning, that this reflects a plastic change of the cortex as a compensatory mechanism to preserve motor function in Alzheimer's disease. So we applied later, after some years, this technique for the study of possible plastic reorganization in other types of dementia. So in this case, vascular dementia. And we found actually that there was no significant difference between vascular dementia and Alzheimer's disease. So both the conditions manifested the plasticity changes, so a reorganization of the motor cortical map. And this was quite interesting in that years, because in that years there was a discussion about the pathophysiology, the possible difference or similarities between the pathophysiology of vascular dementia and neurodegenerative dementias. And there was this trend that we know now that is true, that they share common features. And in these studies, we demonstrated that also from the neurophysiological point of view, the pathophysiology was similar between these two types of dementia. So then we applied the same protocol, the same paradigm to study possible changes in time of this neurophysiological abnormality. And actually, we measured these parameters at baseline and after 1 year of treatment of Alzheimer's disease patients with acetylcholinesterase inhibitors. And we found that there was a parallel, there was a relationship between the degree of changes in clinical parameters, so in the clinical worsening, cognitive worsening of patients, and the amount of long-term reorganization of the cortex. And this is quite interesting because this is a demonstration, this was an early demonstration that the neurophysiology is related to the clinic, so it's clinically relevant and it's not just a study of neurophysiology per se. So, but we know now, after 20 years, that the synaptic dysfunction are a neurophysiological, a hallmark of neurodegenerative diseases. So it's a well-known hallmark of neurodegenerative diseases. And so I will discuss about two diseases, two paradigmatic diseases, neurodegenerative diseases. So Alzheimer's disease, which is clearly manifest with cognitive impairment, and Parkinson's disease, which is mainly characterized by motor impairment. So we know that the original idea of Florinda, which these changes reflect plasticity changes of the cortex, could be tested practically by using repetitive TMS. So there was a technique in years that developed to induce plasticity by TMS. So by measuring the MEP amplitude before and after the induction of plasticity by using some specific paradigms, it was possible to replicate the LTP-like and to induce the LTP-like plasticity or LTD-like plasticity, so long-term potentiation or depression as shown in animal models. And this was demonstrated to be quite reliable because by looking at experimental data in animals, it's also possible to see after the induction of plasticity structural changes in the synapses. So there is here, for example, after a few minutes of these paradigms, growth of a new synaptic button, new synaptic, or an increase in dimension of the already existing synapses. Also, there is a change in the expression of markers of synaptic plasticity, like after the stimulation, there is an increase of GAD65 or of the VGLUT1, that are markers of synaptic plasticity. So it's possible to manipulate, to induce this kind of plasticity. And starting from the original hypothesis of Florinda, other groups started to apply this repetitive stimulation in Alzheimer's disease and mild cognitive impairment. For example, this is related to studies from the Santa Lucia in Rome. The researcher demonstrated that the LPP-like plasticity was impaired in Alzheimer's disease, so in red, so there is no facilitation after the induction of plasticity in these patients. But more importantly, they found that this is true also in the prodromal phase of Alzheimer's disease, in this cohort, so the PROD. In the prodromal Alzheimer's disease also, the patients that had intact plasticity at baseline had a lower rate of conversion to overt dementia compared to the patients that had an impairment of LTP-like plasticity at baseline. And also, this feature reflected and was related to clinical symptoms, so to memory impairment severity. So shifting from the other paradigmatic disease in neurodegenerative condition, experimental studies, and this is from the group of Sapienza, demonstrated clearly that by using TMS is possible to reveal this synaptic plasticity impairment also in Parkinson's disease. So you see here on the left, the black circles reflecting the Parkinson's disease patients in the off condition where there is no facilitation of MEP after the induction of plasticity compared to the healthy subject. And but this is quite relevant, this study, because we measured the cardinal symptom of Parkinson's disease, that is bradykinesia, and we found that the impairment of plasticity was also in this condition clinically relevant, because there was a relationship, a direct relationship between the severity of bradykinesia of patients and the severity of the impairment in cortical plasticity. But in a recent study, we studied also we tested also another paradigm assessing plasticity by using TMS. In this paradigm, it's possible to test short-term plasticity. So it's possible to look at facilitation of MEPs after a short, immediately during a short train of repetitive TMS, and comparing the amplitude of the 10th MEP compared to the first one, it's possible to quantify this short-term plasticity effectiveness. And actually, we found a particular result because in this study, we applied a technique, another technique that was, let's say, artificially able to worsen bradykinesia in patients. But we found that when this technique worsened bradykinesia, here in red, there was no plasticity in the cortex, whereas when the technique did not change bradykinesia, so it was ineffective in worsening bradykinesia. So we saw the emergence of facilitation. So the short-term plasticity processes were intact. And so in this case, for this kind of plasticity, we interpreted this mechanism as a compensatory mechanism, not a causative mechanism of bradykinesia. So putting together this information, by using TMS, we could say that the impairment of plasticity mechanism is confirmed as a hallmark, a neurophysiological related hallmark of different neurodegenerative disease. It is present even in the prodromal stage of this disease, But we don't know yet if this abnormality is primary or is secondary to the accumulation of misfolded protein. This is an open issue. Also, we knew that changes in cortical plasticity are clinically relevant. So it's not neurophysiology per se, it's neurophysiology underlying clinical symptoms. And they parallel cognitive or motor disorders. And also, we know now that different cortical plasticity abnormalities may have different role in neurodegenerative disease. Because some of them could be causative, the causal to the symptoms, and other one could be compensatory. So moving to the EEG, so Florinda was a key player in the very influential line of research about the study of EEG, possible cortical rhythm abnormalities and changes in physiological and pathological aging, which were led by Professor Babiloni and Professor Rossini. In this study, they found that physiological aging is characterized by the reduction in the source of cortical alpha rhythm, particularly in posterior regions, so occipital, limbic, parietal, and temporal. And they speculated that this could reflect the progressive slight decline in visuospatial attention and attentional tone for memorization related to physiological aging. So, but they then studied patients with dementia, with Alzheimer's disease and mild cognitive impairment, and interestingly, this marker of aging was even more prominent in patients with mild cognitive impairment and in Alzheimer's disease. So this data supported the idea that changes in alpha activity in the posterior region support the progression from normal aging to pathological aging. So it's the so-called transitional hypothesis. And these data were really important because they also paralleled cognitive function. So the abnormality of alpha source in the posterior region was related to the global cognitive level and performance in these patients, and also in specific cognitive domains, like in the immediate memory based on focus attention. Also, Florinda was part of this, some important studies demonstrating that this could be a marker also to predict the patients with mild cognitive impairment that will develop Alzheimer's disease in a few years. And this finally led to the approval, I would say scientific approval, that measuring the resting state EEG rhythms could be useful also in clinics, so for clinical trials, for example, to identify specific patients that will benefit from new therapies. So moving on, Parkinson's disease. So Parkinson's disease is another neurodegenerative disorder that is characterized from altered oscillations in the cortex. Starting from the teaching from Florinda, I started this line of research recently in collaboration with Tor Vergata University of Rome, in which we try to find out the markers of early Parkinson's disease by using high-density EEG. So in this study, we found that there are two main abnormalities, and in these studies, we did not measure single sources of EEG rhythm, but we computed functional connectivity in different frequency band and the network-based statistics. So we looked at dysfunction in networks rather than in areas. And we found that Parkinson's disease patients, even before starting treatment, these patients were without any therapy, demonstrated an hypoconnectivity, so a reduced connectivity in the theta frequency band in this network, which correlated with cognitive and behavioral symptoms. But more importantly, we found that the patients with Parkinson's disease demonstrated an increased connectivity in a motor network, focusing mainly on motor cortex, primary motor cortex, and supplementary motor areas, which was strictly connected, linked to the bradykinesia severity. So we knew that from invasive studies, local field potential studies in patients implanted with TPS electrodes, that the hyper beta, so the increased beta oscillations in a wide network, is a hallmark of Parkinson's disease, and it's a biomarker of bradykinesia rigidity. But this study was the first demonstrating that this abnormality could be also found using a non-invasive technique like the EEG, and it's quite reliable and it's really connected, it's really related to bradykinesia. So in a more recent study, so we go on trying to find out whether there are specific network and frequency specific abnormalities in different subtypes of Parkinson's disease. So there is a theory, there is a specific theory in Parkinson's disease related to the pathogenesis, to the pathology of Parkinson's disease and the spreading of the misfolded protein, which dispose two subtypes of Parkinson's disease, the so-called body first, where the synuclein accumulates primarily in the periphery, and the brain first, where this occurs primarily in the central nervous system. And so we recruited a large cohort of patients of the first type and second type, and we found that there was a specific network in the alpha frequency band, which was quite widespread, which resembled the spreading, is compatible with the cholinergic spreading from the brainstem diffusely to the cortex, which discriminate, which is hypo-connected, so that's a reduction in the alpha frequency in this network. This is more evident in Parkinson's disease with a phenotype reflecting the body first Parkinson's disease, then in patients reflecting the brain first Parkinson's disease. And this marker has a high sensitivity in identifying patients with the body first subtype. And this is important because this subtype of Parkinson's disease patients is known to progress more rapidly in dementia and in gait problems. And indeed, we found a significant relationship between the abnormality in this network and cognitive disturbances in this subgroup of patients. So in summary, this data support the idea that along the synaptic dysfunction that we discussed before, also the neurodegenerative diseases share altered oscillation, so can now be interpreted as oscillopathies, in which oscillatory activities are a key pathophysiological mechanism. And also, specific changes in brain rhythms reflect distinct symptoms, and this is pathophysiology, or this is subtypes, it seems. And this is important because EEG markers could be useful as non-invasive and low-cost instruments for early diagnosis or treatment selection. Finally, pathological oscillation can be targeted using specific non-invasive brain stimulation approaches for therapeutic purposes. And in this regard, I will shortly introduce this technique. This is a neuromodulatory technique, the transcranial alternating current stimulation. It implies electrical current with a sinusoidal pattern, and the biological effect of this stimulation is that it entrains brain rhythms at the stimulation frequency. So it's possible to set the stimulation frequency, and it entrains the natural oscillations to the frequency set on the device and over the stimulated area. And so practically it could be possible to boost brain rhythm with this technique. And this is relevant because stemming from the idea that there was a defect, a reduction of the alpha rhythm in dementia in the posterior regions, this researcher demonstrated very recently that patients with dementia with Lewy bodies can benefit from sessions of alpha TACS, so TACS delivered at the alpha rhythm, which boosted the alpha rhythm over the cortical area being stimulated and also improved some specific neuropsychological tests related to the function of the visuospatial function, which is clearly impaired in dementia with Lewy bodies. Another example of this technique is a recent study, quite recent study that we did in Parkinson's disease patients. So we used TACS to boost beta or gamma oscillation on the motor cortex, and we observed that boosting beta oscillation, which are we know pathological, we determined bradykinesia. Bradykinesia worsened in patients. But the interesting part of the story is that boosting the high gamma oscillations, which are known to be prokinetic, we observed really an amelioration of bradykinesia in patients. So it's possible to manipulate brain oscillation. So the final part of the presentation relates to the combination of TMS and EEG. So which are the advantages of this technique? So one advantage compared to the EEG relates to the fact that when we look at the EEG, we are looking ongoing oscillations. So it's like looking at a sea with a lot of waves. So we are trying to search some pathological oscillation during an ongoing activity, which is not all pathological, and so it's a resting state activity. And if we try to correlate the oscillation with a task, we actually can know, we don't know if a motor abnormality, for example, or a cognitive abnormality is the cause or the consequence of the abnormal oscillation. By using TMS instead, we adopt a perturbational approach. So it's like that we throw a stone in the sea and we follow the oscillation produced by the stone. So we know in this case where the oscillations start and we know what to see. So we know where to look at for the propagation of this wave. And this make very more easy to determine a cause-effect relationship also with motor and cognitive symptoms. So the other advantage compared to the TMS in this case is that TMS produces an MEP, but we stimulate the cortex, but from the cortex to the muscle, we have the signal that travel through the cortical spinal tract, the spinal cord, peripheral nerve, and through the muscle. So looking instead of transcranial evoked potential, so that are the potential produced directly over the cortex by TMS, we have no bias of other structure in the middle. And the equipment is quite complicated. So actually is referred to delivering stimulus during the EEG recording with a neural navigation system to match precisely the point that we want to stimulate. And it's possible to do different analysis because it's possible to analyze the data in a power mode. I mean, it's possible to do a power analysis, so to measure the amplitude of these responses, which reflect the reactivity and excitability of the stimulated area. But it's also possible to analyze the data to do a frequency analysis. So, and to look at the so-called TMS evoked oscillation. So it's like that the stimulus given by the TMS reset the activity of the neurons in the area being stimulated, and then we look on the EEG on the natural oscillations produced by the neurons that have been resetted by the TMS. And by this technique, it's possible to understand that to measure not only the excitability of local circuits and also network, but also the connectivity of the area being stimulated and other distant areas. Florinda was really has been really a pioneer of TMS EEG techniques. She developed this technique along with the Finnish colleague, and also she helped in understanding the neurophysiological basis of this transcranial evoked potential. And she worked on TMS EEG for all her career, and this is, for example, on the right, a special issue that we were leading together on the usefulness of TMS EEG in neurodegenerative condition the last year. Together, we tried to elucidate possible biomarkers of aging, physiological aging, by using TMS EEG. We compared transcranial evoked potential in young and elderly healthy subject, and we found that there was, this is the P30, this is a TEP, an early TEP, an early transcranial evoked potential, and we found that there was an hypo-excitability in the sensory motor cortex, and together with an hyper-excitability in the premotor areas after the stimulation of the motor cortex in elderly subjects compared to the young ones. And we interpreted this as a possible compensatory mechanism, so the change in the premotor sensory motor connectivity during aging as a compensatory mechanism to preserve normal movements with aging. And in this study, we also found that there was a decrease in excitability in a wave, the N100, that reflected the connectivity between hemispheres. And this could be a biomarker of the reduction, the slight reduction, subclinical, not clinical reduction with aging of interhemispheric connectivity. This study was a cardinal study. Actually, it was the first study disclosing possible changes in Alzheimer's disease by using TMS EEG. In this study, we found that there was an increased cortical excitability, sensory motor excitability. So these are the dipole of activity after the stimulus of TMS, which is clearly higher in Alzheimer's disease patients compared to the healthy controls. And particularly, this transcranial potential demonstrated for the first time that the hypothesis and the data shown by Florinda 20 years ago of the hyperexcitability of the cortex in Alzheimer's disease were true, were confirmed, and were not biased by other structures than the cortex. And hyperexcitability is really a sensory motor cortex abnormality in Alzheimer's disease. But we found, interestingly, another characteristic in Alzheimer's disease patients, because looking at the number of peak of activity after the stimulus, we looked at a higher number of peaks in Alzheimer's disease patients compared to the healthy subject. And we plotted these waves, and we saw that the dipolar activity was really the same for many tabs, many waves. And so we interpreted that this is a marker of possible disconnectivity of the stimulated area in Alzheimer's disease, like the stimulus stay for longer time in the same areas, it's impossible for the signal to go to other areas because the stimulated area is disconnected to other areas. So it's like a reverberant circuit. And so, because we found these biomarkers, the hyperexcitability and the higher number of bipolar activities, so disconnectivity in Alzheimer's disease, we thought to look at what happens in mild cognitive impairment. So we followed up clinically for 6 years patients with mild cognitive impairment, and we did a neurophysiological evaluation with TMS EEG at baseline, at T0, and we clinically observed patients for years to look at the conversion of patients. So we clinically knew after years which were the patients who converted to Alzheimer's disease and the other that remained stable. And surprisingly, we found in mild cognitive impairment that there was a high number of ways of bipolar activity, like that one that we demonstrated some years before. We then measured a parameter reflecting the synchronization of the area. So as I explained before, it's possible to look at frequency, at oscillation induced by TMS with TMS EEG. And we looked at the synchronization at gamma rhythm, alpha, beta, and gamma rhythm, but we found a decreased synchronization after the stimulation at the gamma frequency in patients with mild cognitive impairment. So there was a disrupted synchronization in this low gamma frequency band. And surprisingly, the most striking result was that when we looked at possible differences between patients that we knew that they became Alzheimer's disease, so here as prodromal Alzheimer's disease, we defined, so PAD, MCI, and patients who did not convert in Alzheimer's disease, we found that these parameters, so the number of peaks, like the strength of each peak and the amount of synchronization in the low gamma frequency band discriminated very well patients that who developed Alzheimer's disease from that who remained stable in years. And this could explain that because we know that there is a disconnection, an early disconnection in patients with Alzheimer's disease. This could be a marker of disconnection and desynchronization of the cortex many years before the occurrence of the disease. Finishing with Parkinson's disease, I had the opportunity to apply TMS EEG when I was in Sapienza with other colleagues of Sapienza, also in Parkinson's disease. And actually, we thought that we found a marker of Parkinson's disease, in particular a marker of bradykinesia, because we found that after the stimulation of the motor cortex, the P30 wave, so the transcranial evoked potential after 30 milliseconds, was reduced in amplitude in Parkinson's disease patients off medication. This abnormality was sensitive to levodopa because it was restored by levodopa administration and correlated with the severity of bradykinesia. So our idea is that this marker could reflect the excitability of the cortico-subcortical network through the basal ganglia and the thalamus and back to the cortex, because we know that, of course, there is the cortical basal ganglia thalamocortical network that is impaired in Parkinson's disease, and by delivering a TMS stimulus over the motor cortex after 30 milliseconds, that we know it's a latency compatible with the arrival of the stimulus to the basal ganglia and reafference of the stimulus to the cortex, back to the cortex. So our hypothesis is that we could really, we have probably a marker of the pathophysiology, a pathophysiological marker of Parkinson's disease, which is non-invasive. And this marker could reflect the altered connectivity in this cortical basal ganglia thalamocortical loop. In summary, TMS-EEG allows the investigation of multiple mechanisms. So it's not just excitability or plasticity, but it could reflect the synchronization of circuits and the connectivity, the abnormal connectivity of circuits. The potential marker that we found in cognitive impairment could be useful for an early diagnosis, maybe for prodromal AD. This should be confirmed in larger sample. And in this figure on the right side that I designed together with Florinda for my residency thesis, we hypothesized in this dotted yellow line that brain effective connectivity changes revealed by TMS-EEG could be useful to identify changes that occur in the preclinical phase of Alzheimer's disease. And finally, the fact that there are distinct patterns of TEPS or TEOS abnormalities in different neurodegenerative disorders. For example, in Alzheimer's disease, we found that P30 is increased, but in Parkinson's disease, we found the opposite. So P30 is decreased. So this could lead to identifying possible biomarkers useful for differential diagnosis for different neurodegenerative disorders. And this was my last slide with some pictures of me and Florinda. On the left, I was very young. I was a student of medicine that was going to graduate. In the middle, there is the photo of my residency. And on the right side, there is the photo of my, this 4 years ago when during my wedding party. So thank you for the effort.
Thanks a lot to Dr. Andrea Guerra for this amazing talk about EEG, TMS, and the excitatory inhibitory balance in the cerebral cortex of Alzheimer's disease and Parkinson's disease patients. So if the people connected to this talk want to ask for questions, please do it by chat. Now the session is open for discussion. I don't know if Angelo or Maurizio
I wanted to make a comment. Thank you, Claudio, and I want to congratulate Andrea, who is, he's young, but he has already something very clear in his mind on which track of research is probably helpful to understand brain functions. And this was Florinda's idea, possibly to detect disease early. So my question would be, I've been historically more involved in studying functional connectivity with MRI, and I'm not that I was, until I met Florinda, I was not that much familiar with changes in cortical excitability, and that could be, investigated as a marker of disease. And so my question would be, if we would predict how helpful is the more neurophysiological approach in the case of Alzheimer, for instance, where we now have more biomarkers available? Is it something which you envision could support or help the clinician? And in clinical diagnosis, we know we have a relatively well-established cognitive tests that could be also considered for population screening. You know, doing investigating brain functionality with EEG or using TMS may be more time-consuming, while maybe drawing some blood may be easier potentially on doing it. So what is how these techniques could be implemented in the clinical assessment of these patients, and which patients probably should be best investigated because they require more a more detailed understanding of the brain physiology in the clinic?
So probably starting from the last question, I think the most suitable patients are the early one, so the early phases or preclinical phases, when we have some biomarkers, like in Parkinson's disease, we have this patient with REM behavioral disorder, so REM sleep behavior disorder, and we don't know if they will develop or not. Probably yes, but we don't know. And in this stage, we could find maybe some marker, because if the disconnectivity of these circuits develop early, very early, we can detect some changes like the one that I show in the TMS very early in the disease, in this population. Maybe also the plasticity processes are impaired early in the disease stage. So maybe we have some clues to say, and coming to the other question, to put together with biomarkers and MRI markers, because I think the multimodal assessment will be the future. Because I mean, we don't have a biomarker that has 100% specificity and sensitivity. And so if we put together some markers, different markers that has like 80% or 85% of sensitivity and specificity, and together we could reach very high sensitivity and specificity. And this is important for patients in the early phase of the diseases, because we know and we hope that we will have some treatments, some disease-modifying treatments for these disorders. And so we have to identify patients that really have the disease in the earliest phase of the disease. But also, I'm really interested, and I did some studies about the responsiveness to treatment. Because by using TMS, we can, for example, we can assess specific circuits and specific neurotransmitters. And we know that some drugs, for example, some drugs used for as an antidyskinetic drug in Parkinson's disease, modulates glutamate, modulates some specific pattern of glutamates. And we have TMS measure to assess glutamates. And we know that some drugs, we have the responsiveness of this drug that is like half. I mean, one patient is a responder, and maybe the one is not. It's like 50% of response in some treatments. It's possible, and it will be very interesting to test whether there is a low-cost neurophysiological test to identify the responders of the treatment, and to identify patients that could respond to a specific drug or not. And so in that way, we could avoid the side effect of the drug in patients that we know that they will not respond to the drug. So this is another practical application of neurophysiology, for example.
Okay, thank you. And Maurizio, do you have comments or?
Sure. Andrea, congratulations, really. It was very well delivered. I liked it a lot. And I didn't know you had done so much on Alzheimer's. So we'll speak more about that. I guess I had the same reaction as Angelo about the, I mean, I guess I have two questions, one more technical and one more clinical. The clinical question is, I mean, if you had like a magic wand, right, and you could get as much data as you want, and, where would you put your money in terms of diagnosis? Would you put it on artificial intelligence and EEG? Or would you put it on, I don't know, drug, TMS, EEG manipulation? Or, let's imagine, dream. Where would you put your money? I mean, if you had unlimited resources, unlimited patients, no problem
maybe,
with money.
but in my mind, I always think that we have to use practical things. So easy to manage, low cost, and very practical. So maybe there are some techniques that are really powerful. Maybe I mentioned TMS-EEG, but actually the dark side of the moon is that we don't know. Still, we don't know. The TMS-EEG responses what reflect what it's really the circuits behind the responses. So it's quite maybe difficult, and there are techniques that are really time consuming, that they need a staff, a very very large staff. Otherwise, there are other techniques that in my mind, I think EEG, for example. So let's
EEG, like standard EEG. If we had like 10,000 standard EEG.
Yeah, because with the recent approach that we have, I was not naive to EEG, but this was not my original field of research in neurophysiology. I came from TMS, so I'm a TMSologist. But looking the results that we have by working on network, not by a single source, so by network functional connectivity in different bands, I think that we could reach a biomarker. Actually, in the first study that we published last year, we found a biomarker that had on 40 patients and 30 controls, so it's not a large number, but it has more than 85 or 90% of sensitivity and specificity in early Parkinson's disease. So I think that putting together
maybe like a Claudio can make a company out of this, like yeah.
Yeah. No,
okay, so yeah,
maybe
I mean,
putting together this measure with other measure, simple measure, like measurement of maybe a single sequence of MRI, just a single one, not too much complicated things, and a simple measure of MRI. We can imagine to put it together, it's possible to detect something that comes very, very, very early in the disease.
So a brief technical question, so I can rope in Camillo Porcaro, who's kind of lurking in the dark there. So EEG got the oscillatory component, which is fine, but the balance, the excitation inhibition balance is actually in the non-oscillatory component. So if people try to, I mean, I know Camillo, of course, has been working on this. And I mean, what do you think of looking at the non-oscillatory component for as a biomarker, or which might be even easier in EEG? Just what do you think of that?
Yeah, I think it's really, really interesting, and because it's possible that some features can be detected only using nonlinear analysis. And we know that nonlinear analysis also reflects complexity of signals. Right. And the network complexity in Parkinson's disease, because you have the complexity of the cortex and the basal ganglia system through thalamus and back, could be really different could discriminate better compared to other measures, the clinical states, or also, who knows? I think I don't know if there are studies, probably not, but maybe even in the very early phase of the disease, there is an alteration in complexity of the signal instead of the excitability or the pure connectivity.
Yeah, thank you so much and congratulations.
And just at the end of this discussion, I want to express my congratulations to you for the great talk again. My comment is that we have with your data and the data by the original work by Florinda, a demonstration that the cortical excitatory and inhibitory imbalance in patients with neurodegenerative diseases can be a key, not only for diagnostic purposes to decide if you have Alzheimer's disease or Lewy body or other diseases, but to have a neurophysiological biomarker of excitatory inhibitory imbalance and probably vigilance dysfunction, where vigilance dysfunction can explain the cognitive fluctuation in Parkinson's disease, for example, but also in Alzheimer's disease, or sleepiness during daytime, the mental fog, the difficulty to concentrate as symptoms, not for the diagnosis, but for the assessment of some vigilance dysfunctions as a symptom, and the fluctuation to drowsiness or light sleep, and something that can be treated if we have neurophysiological biomarkers as endpoints. So thanks for your nice demonstration. And just my final comment is on the body first and brain first Parkinson's disease classifications. This is your data are more related to body first, which means the brainstem abnormalities and neuropathology, because the autonomic symptoms in this Parkinson's disease are related to brainstem abnormalities in the networking, and the ascending brainstem systems are at the basis of the cortical thalamic modulation of cortical excitability and the vigilance and vigilance to drowsiness transitions. So this is a key for the future, to have a neurophysiological TMS, EEG biomarkers of vigilance dysfunctions. But just one comment about the use of TMS. You didn't touch the double TMS paired stimulations at long and short intervals to tap GABAergic or glutamatergic control of cortical excitability. Can you comment on this TMS application?
Yes, thank you for the question. It was a matter of time, but because it's possible to say hours about TMS paradigms, but yes, actually these paradigms delivering two pulses through the same coil are really useful for research, because we know reliably that specific paradigms, so each paradigm reflects the activity of a specific circuit. So we know that we have a paradigm for GABA A, a paradigm for GABA B, another one for cholinergic, for glutamatergic NMDA or non-NMDA, and this was what I was mentioning about the question of Antonini, Professor Antonini, because these are very useful, because we know that some circuits and some neurotransmitters are impaired or maybe are modulated by drugs. We can follow for example, we can monitor the effectiveness of the drug performing the pair pulse paradigm, and combining also that there is an interesting study which was not complete in my opinion, but it has been published in Neurology, which uses different pair pulse paradigm together to classify and to differentiate different types of dementias, so to discriminate, for example, frontotemporal dementia from Alzheimer's disease and from corticobasal syndrome, because we know that these different neurodegenerative disorders affect different neurotransmitter systems and maybe with different timing. And so putting together, we know now that a single paradigm is not particularly useful for clinics, but maybe by combining different paradigms, it's possible to find a pattern. Maybe in a disease there is an abnormality in GABA but not in glutamate, GABA plus cholinergic but not glutamate, and there is another that has a different pattern, and so it's possible to recognize it by using simple TMS protocols. So this is another application, for example.
Thanks a lot and for this discussion and the final explanation. I don't know if Maurizio Corbetta wants to close this session.
Yeah, Claudio, I want to thank you particularly for for putting this together. I think this was very nice, really well thought out. Andrea, thank you for the presentation, and I think this was a great way to remember Florinda. She would be very happy that we're discussing her science and the results, and we're debating how to move forward with science and research. So thank you so much, everybody, for coming and participating.
Bye-bye.
Bye.